All right, so I think we'll probably go ahead and get started as some others kind of filter in. Hello, everyone. Welcome to our Sierra Streams Sierra Science Speaker Series. This is actually the final event in our spring series. I am Jeff Lauder, Interim Executive Director for Sierra Streams Institute, and I'm really excited to basically welcome tonight Lillie Pennington. She's a good friend of mine, and we'll introduce her in just a second. But we wanted to start off with, typically we have a video to play in front of these, but we've been having some technical issues to get that video shared. But just wanted to point out and share that all of these speaker series events that we've hosted for a few years now, and then done virtually this year due to COVID, are just one of the many things that Sierra Streams Institute does to try to basically connect some of the science of our region and of the Sierra to our community. We are a nonprofit organization, so please, if you have some interest in supporting the work that we do, like these types of outreach events, please visit sierrastreamsinstitute.org or reach out to one of us on the Sierra Streams Institute staff to talk about continuing to support some of the research we do and educational efforts. So with that, like I said, this is the third and final virtual Sierra Speaker Series event. In the fall or into next spring, we're hoping to, pending COVID vaccination progress, host these more in person again. Some more news on that as we move towards that in the future. But tonight's talk is actually going to be from a PhD candidate at UC Merced, who is a student of Jay Sexton, who presented last time. And this is Lillie Pennington. She is a PhD candidate in the Environmental Systems program at UC Merced. She received her bachelor's in biology from Oklahoma City University in 2011. And after working as a seasonal intern for the Bureau of Land Management for 2 years, she returned to school to earn her PhD in 2017. She studies the impacts of climate change, specifically drought, on the cutleaf monkeyflower, and the impacts of habitat degradation and fragmentation on vernal pool grass populations. She's also, like I said, a friend of mine and colleague where we work together in the student-run Radio Bioscience podcast. So if you have an interest in science communication, Lillie was the president of that organization. So I encourage you to look up radiobio.net for some cool science podcasts, interviews about biology and those kinds of things. So before welcoming Lillie fully, I just wanted to point out a couple of housekeeping things. Please stay muted for the duration of the talk. And if you have questions, the second the question comes up, feel free to put the question into the chat. And then Alex or myself will be reading those off at the end to Lillie to kind of facilitate a Q&A. Or if you want to hold off on your question and unmute yourself at the very end to ask a question, that works as well. But just hold off on unmuting yourself until the end or enter your question directly into the chat. Other than that, pleased to welcome Lillie Pennington.
Hi, hello. Hang on, I'm going to put my video on, start video. Hi, everyone. Thank you, Jeffrey, for that really great introduction. And thank you for that shout out to Radio Bio. Again, that's radiobio.net for all of your podcasting needs. It's a really great podcast. Yeah, so this is also like the first time I'm giving a Zoom talk. So you might see me like kind of struggling with the pointer, but we'll get through it together. So again, hi, my name is Lillie Pennington. And I'm a PhD candidate at UC Merced, and I study plants. And I'm in the Sexton lab at UC Merced, and as Jeffrey mentioned, you may have seen my advisor's talk last month. He's like a pretty cool guy. And if you didn't see it, that's okay, because this is completely different work. But yeah, we study plants in the Sexton lab. And you may be thinking, like, why study plants? There are a lot of big questions in the world, so why would you focus on plants? And there are some very sciencey reasons why, like, they're primary producers and they're like the base of basically every food web or pyramid or whatever you like. And, they capture carbon, which we're pumping relentlessly into the atmosphere, and they produce the oxygen that we breathe, which is super chill of them. And then they also like filter the groundwater and stabilize soil so we don't have gross water and mudslides all the time. But mostly I study them because they're pretty. And I like to fill my life with beauty, and what's more beautiful than like a forest or a field of wildflowers, right? But I guess it is subjective, but that's why I study them. And I study this little plant here, the cutleaf monkeyflower. And by the end of this talk, I hope to have told you a story of how this plant was affected by the 2012 to 2016 drought in California. But first, I want to talk to you about climate change. So the first thing I'll say about climate change is that we are committed to change. This paper by Morrison and Pincus was published in 2017, and it suggests that even if we had stopped carbon emissions in 2016, which as you all know, we did not, we would still be committed to some amount of change. And you can see here all of these different factors that they accounted for in their model to predict the end of century change in temperature. You can see here with all of these different factors accounted for, we would still be committed to about 1 degree of change. And that's if we had stopped, producing greenhouse gases. And we didn't do that. And so climate change is like a global thing, and we're already seeing these coherent effects worldwide. For instance, warming temperatures that are associated with climate change are linked with changes in precipitation. And this figure shows the percentage change from average during the dry season for all of these different places. But I want to point out the Mediterranean climates, like here in the Mediterranean Basin and the Cape of South Africa, and then here in Southwest Australia and South America, and then of course here in Southwest United States where California is. And all of these places, all Mediterranean climates are expected to have dry seasons as a result of changes in precipitation due to climate change. And the Mediterranean climate is characterized by these cool, wet winters and these hot, dry summers. And it's like an important system because Mediterranean climates are actually home to 16% of plant biodiversity, which is high for a single climate type, and is comparable to rainforests. And so what will happen to the plants that are growing in these systems as they begin to experience drier seasons? Will they be able to undergo the sort of rapid adaptation that they would need to thrive? And in the Sierra Nevada, the end of the century climate change looks a little bit like this, with temperatures on average being 7 degrees warmer, and especially high rates of change in higher elevation areas. You can see the darker color is like a more significant rise in degrees Fahrenheit, and it's like concentrated around these sort of higher elevation areas. And so in Mediterranean climates like California, they're marked by seasonal drought. And so after a while, snow and rain stops falling, and it's just hot and dry. And then in the winter, we have snow falling and some rain. And so during these hot and dry growing seasons, the plants are kind of surviving off of melting snow. And the other thing about climate change, though, is that snowpack is also going to be impacted. And so this graph is showing how much the snowpack will be affected in the future. And this is because as climate continues to warm, we're going to expect more precipitation to be falling as snow. And so with all of these changes in the Sierra Nevada, they're home to thousands of herbaceous plants, with around 400 of those being endemic to the Sierra. And endemic montane species are the most vulnerable to change. And so one of these questions is like, will these plants be able to adapt to changing conditions in the Sierra Nevada? And another thing I'd like to point out is that both of these graphs showed a sort of gradient, with lower elevations seeing less change than higher elevations, and higher elevations seeing a more pronounced warming and a more impacted amount of snowpack. But even without climate change, species experience different climates at different parts of their range anyway. And so on the right here, this is actually the range of the cutleaf monkeyflower, and this dotted line here represents the rear edge, which is the lower elevations, and this dotted line represents the cooler edge, which is the higher elevations. And on the left here, we have this diagram of species range and the challenges that you face at either end. And when looking at like the leading edge and the rear edge, for this talk, the rear edge would be the warmer low elevation edge, because I'm talking about mountains. And then the leading edge is the high elevation edge. And in the Sierra, lower elevations experience higher temperatures and higher drought stress. And then there's this sort of gradient of these two effects up to higher elevations where the temperatures are cooler and there's more water available for longer. And so with these different climates and habitats across the species range, you can also get local adaptation, which is population level adaptation to local climates. And in this classic work here by Clausen, Keck, and Heisey, they qualified the population level differences across the range of Yarrow and California. And along the bottom here, you can see the silhouette of the coastal range and then the valley, and then up to the Sierra Nevada and back down to the Great Basin Plateau on the other side of the Sierra. And so up here on this graph, you can see the different heights of the Yarrow as you move across the range. And so you can really see this like nice gradient of smaller plants in these sort of harsher environments and the biggest plants in like the valleys and the foothills. And not all local adaptation is as dramatic as this. It can be as simple as plants being more fit in the climate that their population is in. And so when we talk about local adaptation, we often talk about fitness, which is simply a measure of the reproductive output of a plant. And the more seeds a plant produces, the higher its fitness. And so using this yarrow example, we can think about a yarrow plant from the Selma population. And at its home population in Selma, it might produce 1,000 seeds. Or in places around it, like in similar climates here, it might produce like 1,000 seeds. But if you take this very same plant and move it up to, say, Tuolumne Meadows, it might produce little to no seeds. And that indicates that in its home climate, it's like more fit and able to produce more seeds. And if you move away from this climate, it becomes less fit. And so that's local adaptation in a nutshell. It's being adapted to your home climate such that if you're moved away from that, you become less fit. And so you may be thinking, if climate is changing and we have local adaptation, then what is happening to local adaptation as climate changes? Well, to deal with climate change, populations will either have to adapt to new conditions, or they'll have to move, or they'll have to face extirpation and extinction. And adapting to change can present in a lot of ways, and a common metric to measure is phenology. And phenology is the timing of developmental stages. And in plants, we think of time to germination, first flower, last flower, things like that. And here we have some of the different developmental stages of the monkeyflower that I study. And here we have this sort of little sprout, and then we have this rosette that forms as it grows, and then it develops into this nice flower. In this graph here on the right from Caradonna et al., it shows the shifting phenology in an alpine plant community in the Colorado Rocky Mountains, with this dotted line here being the average timing of phenology for each of their phenology measurements, which are down here: first flowering, peak flowering, and last flowering. And the shifting more to the left is having earlier phenology, and more to the right is having later phenology. And so the blue shapes here are significant changes from the average, and the yellow shapes are like marginally significant, and then these white shapes mean that this change isn't significant. And so overall, you can see that this community is exhibiting a change in phenological timing, which may be due to climate change. So many of these species are exhibiting a significant change, and they're developing earlier than average. And this may be a result of climate change, because as the growing season becomes warmer, as temperatures change, plants will try to avoid heat and drought by flowering and fruiting earlier. So I mentioned plants either adapting, moving, or facing extinction. And so when I said moving, I didn't mean the plants are just going to look around and pack up their bags and walk up slope when things start to get, too hot. What I mean is their ranges are going to shift. And range shifts have been recorded. And this figure shows range shifting in the Santa Rosa Mountains in Southern California. And range shifts have also been recorded in the Sierra, but I thought this was a sort of nice visual representation of shifting ranges. So in this graph, You have the x-axis, which is the elevation difference from the central of the range, which is 0, the center of the range, which is 0. And then the black circles and the solid lines are those the species and species coverage in 1977. And then these white and dotted lines, that's from 2007. And so in 1977, you can see that the species are further south from the center and are more abundant. And in 2007, you have more coverage here above the central elevation. And so with climate change, you may have range contraction, especially at warmer areas of the species range, where the temperatures become too warm and the populations there start to get extirpated or they die out. And you can also have range expansion, where the cooler areas that were previously uninhabitable for these species become more favorable as climate starts to warm. And so that's how you can have, extinctions and extirpations at the edge of a range, and then you see the ranges shifting higher. And extinctions and range shifts have been attributed to climate change, and that's been known since around like 2006. And so with all this in mind, we come to the California drought. And that drought was crazy, right? You heard all sorts of things like it was catastrophic, it was historic. It was once in every 1,200 years would this happen, things of that nature. And here on the left in this picture, you can see the average snowpack, snow cover in the Sierra. And then on the right, you see the 2015 snowpack, a nice visual representation of how little water we had at that time. And it was really hard, right? But just as we had to live through that drought, the plants had to live through it as well. And so while drought of this magnitude is unlikely to happen again in the near future, we are likely to see hotter temperatures and reduced snowpack, just as we did in the drought. And in this way, the drought sort of serves as a natural experiment of what conditions might be like in the future. And so using the drought and these factors of plants that are all impacted by climate change, my dissertation research seeks to address these questions. How are plants responding to significant climate events? And response here means both changes in phenology, but also changes in fitness. Did plants quickly adapt to the drought? If plants can adapt quickly to significant events, then perhaps the variation that they need to respond to future climate change is already available in these populations. And are responses similar across the species range? Or is there a part of the range that's less likely to be able to respond? And will we see more of those contractions, or will we see range expansions? So I'm addressing these questions in this precious little plant, the cutleaf monkeyflower. It's Mimulus laciniatus or it's Erythranthe laciniata. I've heard it both ways. There's a little argument over it. It means this plant, the cutleaf monkeyflower. And the cutleaf monkeyflower is a great plant to study these sorts of questions in because it's an annual plant, which means that you can measure its lifetime fitness easily, the amount of offspring it can produce in a lifetime, because it just needs one growing season. So at the beginning of the growing season, you can measure all sorts of things, and at the end of the growing season, you'll have its lifetime fitness because it will die. And it is a Sierra Nevada endemic, and it's restricted to the western slopes of the Sierra Nevada. And it grows in these exposed granite outcrops in these mossy snowmelt seeps. So it will be directly affected by the amount of snowpack available in the future, because that's where all of its water comes from, snowmelt. And it's 95% self-fertilizing, so it doesn't outcross very often. But again, most importantly, it's very cute, easy on the eyes. And so another reason why the monkeyflower is a great species to answer this in is because it's been questioned whether or not endemic and self-fertilizing plants will be able to keep pace with climate change, because endemic plants tend to have smaller populations and they have smaller ranges, and self-fertilizing plants don't have the same access to genetic recombination as outcrossing plants do. So will this plant be able to adapt quickly enough to climate change? No, with all of that in mind, it doesn't have the variation necessary. And so how can I figure out how this natural year-long drought affected native populations of this monkeyflower? And how can I tell if the plant did adapt quickly to this change? And so my lab performed a study. It's called a resurrection study. And Resurrection studies are really cool. You may have heard about plants being grown from seeds harvested from deep in the sediment of lakes or bogs or in the Arctic tundra, or like this study here where an ancient date palm that was actually extinct was resurrected from a 2,000-year-old seed. And resurrection studies are really cool because when you think about it, seeds are sort of like time capsules. They contain the genetic code of their parents or parent in the case of self-fertilizing plants. And in growing these old seeds, we can see what plants from a long time ago looked like, even what plants that are now extinct look like, and what sort of phenotypes may have been prevalent a long time ago. And when I say phenotype, I mean basically what the plant looks like and also the timing of its phenology. So days to first flower is a part of the plant's phenotype, as well as things like height and the color of the plant. So in the case of this ancient palm, we're looking at the phenotype of a 2,000-year-old plant that is actually now extinct. And resurrection studies use these ancient seeds as well as contemporary seeds to measure change over time. And my resurrection study is similar, although my seeds are certainly not as spectacularly old. So this is a sort of diagram of my resurrection study, like where the seeds came from. And so this graph is kind of busy, but what I want to point out is this black line, which is the measured snowpack of that year. And so my study is using seeds from before the historic 2012 to 2016 drought to measure change in response to the drought. And you can see my collections here in 2005 where we had really high snowpack, and then in 2014 here where we had super low snowpack. And growing these collected seeds can show us what the phenotypes were more prevalent. We can see what phenotypes were prevalent in 2005 as compared to what phenotypes were prevalent in 2014. And so we can see if there is a difference in the phenotypes and if there was a certain type of plant that grew better in 2014 at the height of the drought. And so to measure this, we grow these seeds in common conditions and we measure the difference between them. And so we grow the plants in these growth chambers in average conditions and in temperatures that we found that they grow best in and with plenty of water. And here you can see the plants in their little trays that I put them in the growth chamber. We've got little rosettes here on the right, and then a more developed tray on the left with some flowers and bigger rosettes. You can't hardly see the flowers, but they're there. And in growing them in these common and cushy conditions, we can see what the genes in these seeds hold and what phenotypes they produce. And so we grow these plants in these nice conditions and we see if there's any difference between the two generations, the drought and the non-drought generation or the pre-drought generation. And so this is work from a former master's student in my lab, Erin Dickman, and she was interested in measuring the rapid adaptation to drought, meaning that during the drought, was there a measurable and significant change in population level genetic variation? And in these graphs, we have the elevation here on the x-axis and mean days to emergence on the left and mean days to first flower on the right. So we're looking at the phenology difference between the two generations. And the black circles are the pre-drought generation and the gray triangles and the gray line is the drought generation. And both days to emergence and days to first flower were significantly different between the generations. And for days to emergence, you can see that the difference is dramatic in the higher elevations. And the higher elevations also flowered later in the pre-drought generation. So now the question is, are these changes drought adapted? And I have like two approaches to answer these questions. So I have a field study, which is a common garden experiment where I take, hang on. I also want to point out that the drought generation has a reduction in variation. And so with this reduction in variation, do we also see an impact on local adaptation? And so again, I have these two studies. I have a field study, which is a common garden experiment where I take populations from all across the species range and I grow them together in these 3 different gardens that are in 3 different climate zones across the species range. And then I also have a growth chamber experiment where I grow plants from across the range, but I grow them in an environment that I can control. So for my field experiment, I have these questions. Does earlier emergence and flowering confer increased fitness in contemporary conditions? Because we saw earlier emergence and flowering in the drought generation. So in these contemporary conditions, growing them out just in the wild, is there a difference in their fitness? Does drought affect local adaptation? When we see a reduction in variation, does that affect local adaptation? And are we seeing range shifts? Are lower elevations less favorable than higher elevations? And so I work with 9 populations of the cutleaf monkeyflower. And this is a map showing where they are and their elevations. So just to orient you on this map a bit, there's Hetch Hetchy up here. And then Yosemite National Park is like, you know, this sort of area here. And if you're familiar with the area, this is actually on the way to May Lake. This population is off the trail on the way to May Lake. And then down south here, this is outside of Shaver Lake. And this population is like right before Aubrey on Highway 168, and this is around Bass Lake, so on and so forth. And so these populations with the little houses are my source populations where we collected seeds to use in the study. And the stars are also source populations, but they're also the gardens. And we have the low garden and the high garden here. And they represent the most peripheral, or some of the most peripheral populations. And then the middle garden is most central. And it's actually kind of like the most central of the whole species range, geographically and elevation-wise. And at each of these 3 gardens, I have 30 individuals from each of the 2 generations, the drought and the pre-drought. And then I have all 9 populations represented for a total of 500 individuals at each garden. Or at least what I hoped would be 540 individuals at each garden. And so this is a picture of my little trays out there. This is, I put all of the seeds in these little trays. And this is a tray that's ready to be harvested, all the pretty monkey flowers. And the trays, I put the trays out in the winter time and they germinate in the wild. And then once the growing season starts, I begin to make weekly visits. And I measure phenology. And I have a number code for the stages of phenology. For instance, 1 is like germinating, and then 5 is flowering, and 7 is senescing. And then there's like numbers in between. We don't need to know all of them. And once the plant is senescent, I measure the number of fruits, and then I harvest the plant. So I also want to share with you all some of the harsh realities of fieldwork, because sometimes you get to a site and your trays just perfectly flipped over from where you put it. And sometimes they've been crushed and they're overgrown. And sometimes your trays have been straight up pooped in, and these things happen. And they actually happened quite a lot with this study that I'm sharing with you guys. And it's okay, because the study was actually more of a pilot study. I, of course, had hoped for good results, but I always planned for a second study. And I do have a second study out in the field right now, and I'll be collecting the data probably through August. And it's already more robust. So I'm really looking forward to the rest of the season. But I do have results from this pilot study where I had all of these things happen to me. And so for this pilot study, I didn't have very high survival. And in fact, I had to remove the high garden from my analysis because the trays were overgrown, like in this middle picture here. So here are some of my results. When I look at fitness across the different gardens, on the x-axis, we have the low, middle, and high garden. And then on the y-axis, we have fruit count. So we use fruit count as a proxy for fitness, because how many fruits a plant produces is an indicator of how many seeds it will have, which are the offspring. And so the lighter blue color is the drought generation, and the purplish color is the non-drought generation, or the pre-drought generation. And you can see that there is a difference in the central elevation garden, the mid-elevation garden, but overall, the differences are not significant. And here's a visualization of local adaptation. And here on the x-axis again, you have the low elevation garden, the middle elevation garden, but I didn't include the high garden because the survival was so low. And to see, so this is, I'm looking at local adaptation here, and to see local adaptation in action, you would want to see, for instance, in the low garden, you would want this blue dot, which is the low populations, to be higher than it is in the middle garden, the central garden, because that would indicate that these plants are more fit in their home garden than they are when you move them away. But we don't see that. In fact, we see all populations performing better in the middle garden. And in fact, we have a higher signal of adaptation in the drought generation middle elevation populations than we see in the pre-drought generation. So it's difficult to conclusively say what happened to local adaptation based on these results, but it is looking like across generations, the middle elevation garden is better for these plants. So back to my questions. Does earlier emergence and flowering confer increased fitness in contemporary conditions? Fitness was not significantly different between the two generations, but the contemporary conditions were actually a slight drought, so pretty close to average. So maybe it's okay, or maybe it's to be expected that they didn't have a huge difference. And then, does drought affect local adaptation? Lower populations are not showing signals of local adaptation, but it might be more of a climate change result than the drought affecting these populations. And are low elevations less favorable than high elevations? It's hard to say without the high garden data, but all populations did perform better in the central garden. I'll let you guys think about that for a second while I take a drink of water. Okay, so again, in prior work, we found that we have the drought generation emerging and flowering earlier. And so that leads me to my next question. Are these changes actually drought adapted? But to address these questions, what I'm going to do, I did a growth chamber experiment. And we have these growth chambers where we can control temperature and light and water. And here's a picture of some of the monkey flowers growing in our growth chambers. And I mean, they're out of control, right? We can really get them going. So carefully controlling the temperature and water, I wanted to see how these two generations, the pre-drought and the non-drought generation plants, do in drought conditions. And so my questions for the growth chamber experiment, does earlier emergence and flowering confer an increase in fitness in drought? And how does drought affect populations from different climates? And so the methods for my growth chamber experiment, I wanted to look at drought, but I wanted to look at drought in two conditions, in average present conditions, and then in the future warmer conditions that we're expecting to see, the 7 degrees increase that we're looking to see at the end of the century. And so for the daytime temperature, I was growing the plants in 23 degrees Celsius, and then in the present average conditions, and then in the future warmer conditions, I was growing them in 30 degrees Celsius. So this is like my hot treatment, and this is my average treatment. And so I have two more treatments. In each of my hot and my average treatments, I have early and late drought. And so the early drought is meant to simulate a sort of like catastrophic drought. So I withheld the water at 16 days. And the late drought is sort of meant to simulate the usual drought, the seasonal drought that you see in a Mediterranean climate. And so I withheld the water after 35 days. And so for this, I had 10 families from each population for each generation. And I had three representatives per family. And then I have all of those in each treatment. And so for the whole study, I had 2,160 plants. And this is a picture of me checking my plants. And you can see I'm wearing a mask because I did this during COVID times and I followed the rules. And so I was going to campus every day and I was taking phenology measurements, and I took days to emergence, days to first flower, days to first fruit, and days to senescence. And then I also took the height at first flower, and then I would take the final height after the plant had died, after the plant senesced. I would take the final height, and I would harvest the fruits, and I'd measure the fruit mass and the plant biomass. And then my measurement for lifetime fitness is total flower, total fruits. So like how many offspring did the plant produce? And so these are my phenology results for my growth chamber experiment to see if we still have the same difference as we saw in the Dickman study. And with the different treatments, the phenology data isn't as clear-cut as the Dickman results that I've shared with you. So this graph is just the average non-drought treatment in the growth chamber experiment as this treatment is basically the same as the methods for our breeding generations. And the differences in germination time are similar to the Dickman study, and the two generations are significantly different. And I don't have my first flower graph quite yet because the story is a little more complex than emergence, but we're seeing similar patterns to the Dickman paper where we see higher elevation drought plants flowering earlier and with a huge decrease in variation, and then this huge variation in the pre-drought generation. And so here we have the fitness results of the growth chamber experiment, and using a regression analysis, I found that generation does have a significant effect on fitness, although you can see that's not a huge magnitude. And the drought generation plants, I will also point out there is a significant difference between treatments. And in the hot non-drought treatment, there is a huge reduction in thickness. And so that suggests that there's something about a hotter climate that these plants are being affected by the heat in such a way that their fitness is reduced even with ample water. And so this is a picture of my trays, some of my trays from this experiment. On the far left, we have the average temperature non-drought treatment, and then this is the average drought, and then this is the hot temperature non-drought treatment, and this is the hot drought. You can see that in both treatments, both of the temperature treatments, the hot drought was pretty catastrophic, and you can't hardly see any plants growing in the trays, right? But they are there. And they're there, but they're just like super stunted. And at this point, they're kind of dying. And they've all been growing for the same amount of time when this picture was taken. And then here are the two non-drought treatments next to each other, with the average treatment being here on the left and the hot here on the right. And these trays, again, they've been growing for the same amount of time. And just visually, you can see that the average temperature treatment was really thriving in comparison with the hot treatment. And so in this graph, we have population on the x-axis, and I aggregated the populations into low, mid, and high elevation pops. And on the y-axis, again, we have fruit count. And looking at this graph, there are a couple of patterns. The low elevation plants are out for—
Hey, Lily?
Yes.
Sorry, I apologize. I was going to say, I don't know if you want to maybe turn off your screen. The slides are lagged by just like probably about 10 seconds or so right now.
Okay, let me turn off.
Sorry, thank you.
Wait, now I have to find out how to get my pointer back on here. Okay, hang on. Am I doing it?
Sorry.
Oh my gosh, I really can't figure out how to get my thing to close. Oh wait, if I turn this off, then does it work?
If it's too much of a hassle, don't worry about it. I just noticed the slides were a little bit behind.
Oh, it happened. Someone did it for me. Thank you, whoever did that for me. Okay. What was I saying? Oh, okay. I aggregated the populations into low, mid, and high elevation populations. And then on the y-axis, again, we have fruit count. And so, yeah, I'm seeing a couple of patterns. The low elevation plants are outperforming the high and mid elevation plants in every treatment. You can see that. And then potentially, I feel like that potentially means that our average conditions are already too warm for the high elevation plants, which doesn't really bode well for them at the end of the century. And there's not a lot of difference between the two generations, but it is a significant difference statistically. And I'd like to point out that in both non-drought treatments, the drought generation outperformed the non-drought generation in the high elevation populations. And in the mid elevations, sorry, gotta use my pointer. And then in the mid elevation populations, the non-drought generation outperformed the drought generation plants in each treatment. So if it's me reading the signs, drought generation plants in the high garden may be better equipped for the future. This would suggest that the variation necessary is there to rapidly respond to heat events at least. And in the central garden, the drought may not have selected for the plants that are best equipped for hotter climates. So the changes may have actually been maladaptive. And why that is, I'm really not sure. But again, the difference is like super slight. So only time will tell. And so for my questions again, does earlier emergence and flowering confer increased fitness in drought? Yes, a little bit actually. And how does drought affect populations from different climates? Well, the high elevations can't really hack it, but also no elevations can when it comes to drought. And even though my drought treatment was very catastrophic, the almost all of the populations just couldn't hack it. So the overall conclusions for my study, in my field experiment, we saw that local adaptation is potentially affected and the generations do perform similarly in contemporary conditions. But again, there was a slight drought, so it may be unsurprising that the two generations actually perform similarly. And then in my growth chamber experiment, we have lower elevations outperforming higher elevations, and there is a benefit to being from the drought generation in drought. But as we see a different, as we see the lower elevations doing better, climate adaptation may be more important than adaptation to drought in the future. And this is my takeaway slide. And I think my main takeaway is that we can study evolution in real time. So studying populations year to year and finding population level difference is studying evolution in real time. And since evolution is a change in trait frequencies. And in studying these differences, we can look at the potential for adaptation to future changes. It's pretty cool. And then I'm also interested in what's going on in the monkeyflower seed bank. And I've collected some soil samples this past growing season from where the plants grow. And so this summer, I'm hoping to do a growth chamber experiment, germinating whatever seeds are in the soil and seeing if there's some drought resistance there in the seed bank. And I'm also interested in what you all think. So, I don't know, put in the chat box, like, what plant would you like to resurrect? What questions do you think that we can answer with these sorts of studies? And then, of course, do you have any questions for me? And then I also have this acknowledgement slide. And I have my wonderful lab here. It's the picture of a few of us out doing some monkeyflower work. And they give me such great feedback and support of my work. And then I have these undergrads who help me with my work, and you can see me and my undergrad Mimi and Sunshine. They're long gone, they graduated, but they did help me a lot. And then there was also Michelle who helped me with lab work. And then of course, my wonderful partner Candace, who despite being a microbiologist, hooked it with me across the Sierras as my data scribe. You can see her here out at one of our my high garden. She's diligently taking notes. And then my funding, the UC Merced Fellowship that funded my first 2 years, the Eugene Cotto Robles Fellowship, and a handful of professional development fellowships. And then I was granted a Matthias grant through the UC system for my fieldwork. And up here, you can see my dog Billy, who is actually more of a hindrance to my work than anything, but he is super cute. So yeah, I'll be happy to answer any questions that you'll have. And thank you so much for listening to my talk.
Thank you so much, Lily. That was awesome.
Was it dope?
Yeah. So I said earlier, basically there's kind of two ways to ask questions. You're welcome to put your questions into the chat, and then Alex or myself will kind of DJ them to Lily. Or if you do want, would you rather ask a question actually verbally yourself, you are welcome to raise your hand, and then we can call on you. To raise your hand, if you click on in your Zoom screen reactions, you can click raise hand. Or if you can't do that, can't figure it out, feel free to just say I have a question in the chat, and then Lily can call on you that way, or one of us can.
I actually would say I cannot click on anything in the presenter I'm in, so.
Can you see the chat screen right now?
Can I do what?
Can you see the chat screen?
I cannot. I can't figure out how to click it, Jeff.
Okay, so yeah, we'll go ahead and kind of just DJ these to you, and maybe Alex and I can alternate.
Okay.
So the first question is from Dan Webster, and he asks, well, first he says, wonderful study, and asks, are there others doing similar work with other plants in the Sierra?
Thank you. Thank you so much, Dan. Yes, there are others doing, there's actually a resurrection initiative, and it's actually not just in the Sierra, it's kind of nationwide and also worldwide, where people are collecting seeds, or they're finding that they have these seed collections. And it's this great resource for experiments because, it's like a natural experiment. Climate change is changing these populations, and you can look at that change using these older seeds. And off the top of my head, I can think of a mustard resurrection study that is nationwide. And that's like the Frank Lab, which I think is in North Carolina. And then there's other monkeyflower work being done in the Sierras with Mimulus cardinalis. And that's the Shima Safe group. And there's also Mimulus futatus work being done at Berkeley. And Of course, there's also the other sorts of studies where the seeds are much older. Seeds are being collected from the sediment under lakes and stuff. And I can't tell you exactly who does that work, but it is being done. Yeah, thank you.
Okay, another one from Shane. Do you have any idea for how long E. lacinata seeds remain in the seed bank? Might wet and drought years produce and deposit different phenotypes into the soil, which then may germinate depending on the subsequent year's weather?
Yeah, for sure. And I think that's what makes a seed bank study so interesting. So to answer the first question, no, we don't actually have a very good idea of how long the seeds will last in the seed bank for Erythrantholus lineata. And yeah, so we don't know how long. And for sure, there are different phenotypes that are being deposited into the seed bank for each year. And I think that's what makes it interesting, because you might have this huge wealth of variation in the seed bank that might be able to respond to future different conditions. They might germinate and be able to, and make sure the population continues to thrive. And then, but conversely, you know, the variation might also not be there in the seed bank. And so I'm kind of looking forward to seeing what variation exists in the seed bank, and if we see more drought adapted plants or if we see more non-drought adapted plants. Thank you. Thank you for asking
that. Alan Kai, who I think many of us know, hi Alan, asks,
Alan.
what is an example of an improvement to your experimental setup in the field that you made between your pilot study and your current project?
Ooh, Alan, thank you for that question. What did I do differently this time? Well, so I actually, I had to change two of my gardens, my middle elevation garden and my high elevation garden, because the middle elevation garden is inaccessible because it burned down in the fires last year. And then the high elevation garden is really far away, and it was hard to get to and visit a lot. And so one of the changes I made is I changed the high elevation garden to a lower elevation, but still high elevation garden. It's near Tenaya Lake in Yosemite. So I'll be able to easily visit that garden more often and hopefully get some better data from there. And then in my previous study, or in the study that I shared with you all today, I kind of had these different quadrants at each garden where I was putting trays out in different sorts of environments because I didn't know which areas would be best for trays. And so this time around, I had a better idea of where water was going to go and where the plants would grow better in their little trays. And so I think I had better placement this time around. And I'm already seeing a difference I have a lot more survival at the highway garden. And I had to change my middle garden. As I said, it's not around Turtleback Dome in Yosemite. And I also have a lot more germination and survival there as well.
Can I just quickly tack on just an additional question to that? So I'm curious, have you tried any like exclusions for like the wildlife scat situation, or is that just not feasible? Or like, are there other methods that you could use to at least alleviate that particular issue?
Yeah, no, I did. Well, because the plants I mostly put the trays out on these like granite slopes. And so to like put up a fence or something, it would have been really cumbersome, and I didn't do that. But so it was more just like a wing and a prayer. I was really hoping that I wouldn't have like a lot of interference from animals. But in my previous study, I put that picture up, but that was the only tray that had poop in it. I just thought it was funny. So there wasn't a lot of interference from animals in that study. It was mostly like trays getting flipped over, probably because of snow flipping it over. Snow moves down the slope, it can flip things over, and yeah, things getting crushed under the snow at the high garden and at the middle elevation garden. And I did change my tray type. So, because it seemed the first trays that I used were really flimsy. You could just like crush them in your hand, which I didn't even think too much of the first time I did it. But the second time, Jay gave me some of his old trays, which are just made different, and they're like so thick and sturdy. So I didn't have as much crushing worries. for this study. Yeah.
Ray asks, would the flowers adapt better if the drought had a much slower transition?
So like, it's kind of like, are plants going to be able to adapt as we slowly get into drier conditions rather than having to adapt to this like snap drought that lasted for a really long time? And I think for these self-fertilizing plants, let me think about this. Would a slower drought, I think more drought adapted phenotypes would be able to build up in the soil if a slower drought was happening, whereas in a quicker drought that we saw with the 2012 to 2016 drought, which was kind of like a snap, and then 5 years we had no water. Well, it was kind of like that. There was still snowpack melting, but then, you know, at the height of the drought, there was really not a lot of snow available. I think there, the plants had to rely, or the populations had to rely more on the variation that was already present. Like if there happened to be a drought adapted phenotype already there in the population to like to keep the population moving forward into the next drought year. Yeah, I think like a slower move into like higher drought or more pronounced drought conditions. It might help the populations like continue to thrive and build up the variation that's necessary to respond to drought. I think so, but that's a great question, and I'm going to think about it more.
Good question for future research.
Yeah, exactly. I'm like writing it down.
So actually what follows is just a bunch of comments of praise. So thank you for devotion to our environment during these difficult and changing times. Fascinating talk. Best of wishes to you.
Thank you so much.
And see you on the trail from Dan Webster.
Oh, for sure.
And I actually had a couple of quick questions. So one is that in your kind of common garden experiment, you showed that the mid-elevation garden did best. So high elevations did well in the middle garden. Low elevations did well in the middle garden. I just think, I'm curious if you can, I know it's kind of conjecture, but talk a little about why you think that might be happening. So is that, because we typically expect under climate change in mountainous regions that as we have warming, lower elevations are going to be pressured to move or go extinct or adapt. But higher elevations that are stressed by the cold and maybe more moisture are actually kind of starting to come into perfect conditions. So why do you think you kind of saw a little bit of this almost range contraction where populations on both sides didn't do as well, except in that middle garden?
Yeah, well, I think a lot of it, honestly, Jeffrey, can be explained by the low survival that I had. Because if I had my high garden, then we might see there might have been like a difference in the central garden, like maybe doing better, or the central populations doing better in the central garden than they did in the high elevations. But we might also have seen like the higher elevations doing like a lot better in the high garden. And actually, some prior work in my lab, which was done a while ago, Jay, for his dissertation research, he did a common garden experiment, and he did see a signal of local adaptation for the low gardens where they did perform better, in the low populations where they did perform better in the low garden, and then they didn't perform as well in the central garden, but then they actually performed really well in the high garden. And then in that experiment, the high garden plants performed really poorly in the low and central, or the high population plants, excuse me. The high population plants performed really poorly in the low and central gardens, but then they did very, very well in the high elevation gardens. And then that had the highest fitness of all of the whole study. And so if I had seen a similar pattern in my study, then the high elevation plants that I included in my study would have had the highest fitness in the highest garden. and they would have had the highest fitness throughout the whole experiment. And so I think it's hard to say without that data from the high garden if we're going to see more favorable conditions in the high elevations, which we did see before in Jay's study. And so I think it's a signal that the low elevation plants did better in the central garden than they did in the low garden. That might be a signal that we might see some range contraction, because in Jay's study, the low elevation plants performed better in the low garden than they did at the central garden. And so we might be seeing some range contraction there, but it's just difficult to say without the high garden data. But it was really like, that garden was decimated. Like, I had maybe 5 plants that grew. So hopefully I can get some better data this year from this pilot, or from this study that I'm doing.
Yeah, I just now realized that I kind of conflated 2 of your figures, and that you did not have a high population in your garden experiments. So that helps explain it, yeah.
Yeah.
Alex actually had a question that she wrote into the text. Alex, I don't know if you just want to ask it yourself, or if you would like me to read it.
I can ask it. I just wanted to get it down. Lillie, what are your thoughts about conservation efforts planting like local endemics that may not be able to handle where that area is projected to go with climate change, versus planting species not from really that area, but in places that have a similar climate to what the original area is like projected to be, if that makes sense.
Like assisted migration, like moving plants, for my plants, it would kind of be like moving plants upslope, like sprinkling seeds across the granite slopes, or like moving plants from a different population up to a more favorable population. What do I think about that? Sometimes, I go back and forth on it, because sometimes I'm like, what can we do? Like, this change is happening really quickly, and it's because of carbon dioxide emissions and greenhouse gas emissions that are because of the way, human populations are living these days. And so does it also make sense for us to move those populations upward to help them survive? But then I'm also like, well, extinction is, natural, and it happens. And there are plant species that are doing well under climate change too. And if they become more prevalent, is that really a big problem? And it's just, it's just kind of like how you look at it. If you want to preserve what we have now, then we might want to go with assisted migration. But if you're, if we're just going to accept what happens, then then we have to accept that some plants will go extinct. Yeah, I don't have like a really hard and fast opinion on it. I really go back and forth. So, yeah.
I think like most things in conservation management, it depends on your goal. That's kind of what you're saying.
Exactly. It's kind of like how the different land management systems in the federal government like have different goals. So in the parks, where you're trying to conserve and protect what we have now, then maybe assisted migration would be something that they're interested in. But then like in the other land managing agencies, like the Bureau of Land Management or the Forest Service, where you kind of have more resource use, I don't know, maybe they'll have different approaches to these changing populations. Yeah.